Hello, I'm Dr. Eleonore Mira, and I'm happy today to talk about the use of legume trees for soil bioengineering in the Caribbean, which represent ways for conservation and restoration of riparian forest. Riparian ecosystems are a small area with a disproportional ecological importance under pressure because of the increasing human activities. Conventional water-based management practices use hard technique based on pure or concrete riprap, leading to the degradation of riparian ecosystems. So today we need new cost-effective and sustainable solutions. Soil bioengineering is one of these solutions. It can be defined as the use of plants as building material in works. The selection of suitable plant species is therefore a first requirement in the implementation of soil bioengineering techniques. Nitrogen-fixing legume species are particularly relevant. However, the propagation by cuttings of native Caribbean legumes is difficult, and we lack of knowledge concerning their germination and establishment phase. The study was conducted in Guadeloupe, located in a biodiversity hotspot. The natural riparian ecosystem is forest, and there's 20 native legume tree species potentially useful for soil bioengineering, which is currently developing in Guadeloupe. But we need to acquire information on these species to use them. The aims of our study were to provide practical tools for the use of Caribbean legume tree species in soil bioengineering. We selected 5 Caribbean native legume tree species, and we measured germination rate, survival, herbivory, and some morphological traits like stem length, root diameter, and the biomass of each compartment. We selected low-cost experimental conditions. Seeds were sown in a mix of river sand and natural soil under a shade house and automatic irrigation. Seedlings were left 3 months in pot before excavation. The selected species presented different ecology and fruiting phenology. High germination rates were reported for all the species, and a remarkably high survival were also reported. At 3 months, 100% of the planted seedlings survived for all the species. A high sensitivity to herbivory were reported for Lonchocarpus heptaphilus. A principal component analysis revealed differences between species. Lonchocarpus heptaphilus and Inga ingoides were the species with the lowest growth and the lowest root investment. In the case of Lonchocarpus heptaphilus, its high sensitivity to herbivory make this species not very interesting for soil bioengineering. Inga ingoides can be suitable to increase the diversity in works. Lonchocarpus roseus is a critically endangered species in Guadeloupe. This species presented a fast development of its shoots, but a low development in its root system. This strictly riparian species remain suitable to increase diversity in soil bioengineering works and should be favored for its conservation. Pterocarpus officinalis, a swamp forest species, presented the fastest growth and invested as much in the growth of its shoots than in that of its roots. The use of these seedlings is very interesting in soil bioengineering in brackish area. Inga lorina also presented a high growth rate and invested in its root systems. The use of these seedlings is very interesting for soil bioengineering to restore riparian seasonal evergreen forest and rainforest. Seedlings of some Caribbean legume tree species presented characteristics suitable for soil bioengineering. Some of them appeared interesting to increase the diversity on works, and others presented a fast root development and a fast growth, particularly useful in soil bioengineering to retain the soil. Soil bioengineering can be a tool for conservation of some species, such as Lonchocarpus roseus, and beside the aim of this study focused on the restoration of riparian forest, some selected species are not strictly riparian and strictly Caribbean, and can be used in larger forest restoration in the neotropics. Thank you for your attention and good conference.